Today I'm going to be talking to you about stem cell research and stroke. According to the World Health Organization, stroke is characterized by the loss or death of brain cells due to a lack of oxygen and blood supply to the brain. Transient ischemic attacks, also known as mini-strokes, as they usually are a warning sign relating to a mild to severe stroke. These occur when there is a temporary blockage in the blood vessels of the brain, resulting in a temporary loss in blood supply. Ischemic strokes are the most common kind in where an artery to the brain becomes blocked by a blood clot, preventing blood circulation to a specific part of the brain. There are two different types of ischemic strokes. Thrombotic strokes refer to the buildup of cholesterol plaque in the arteries, preventing blood flow to the brain, whereas embolic strokes refer to a blood clot from a different part of the body traveling to the brain where it becomes stuck in a blood vessel, causing a stoppage in blood flow. Hemorrhagic strokes are the final type of stroke in which there is a rupture of the wall of a blood vessel, causing blood to leak throughout the brain. There are also two types, including intercerebral hemorrhage, where an artery ruptures within the brain, and subarachnoid, where there is bleeding on the surface of the brain. So what are the effects of stroke? The effects of stroke vary for each individual depending on multiple factors, including the general health and activity of the individual prior to the stroke, type of stroke, location and size of the blockage or rupture, and which area of the brain is damaged. The frontal lobe controls reasoning, motor skills, personality, language, and cognition. The parietal lobe controls processing senses such as touch, pressure, and pain. The temporal lobe controls memory and language, including hearing. And finally, the occipital lobe interprets and processes vision. So then, what are stem cells and stem cell research? Stem cells are unspecialized cells, meaning they have no specific function, however, they can potentially transform into any specialized cell. Therefore, these cells have potential and can be quite useful in the medical world for tissue regeneration and repair. These stem cells can help in understanding the brain as well as the creation of medicine that could stimulate the repair mechanisms in creating new stem cells to repair the damaged ones. Another avenue in research involves the growing of stem cells in the lab to inject into the blood to stimulate growth and repair of the damaged parts of the brain. Embryonic stem cells are those stem cells derived from the inner mass of a human embryo and are pluripotent, meaning they can differentiate into other cells within the human body and replicate indefinitely. Initial research saw these cells injected into rat brains with stroke lesions, which depicted survival and differentiation into mature neurons with improvements in structural repair and functional recovery. Further studies found the survival and formation of new nerve cells as well as productions of neurons which could make connections with existing neurons. Advantages to this research included improvements in motor and sensory functions, as well as reduced infarct size. Disadvantages included formation of malignant tumours and ethical concerns surrounding embryo use. Inducible pluripotent stem cells. 2007 researchers developed a way for mature human cells to resemble pluripotent stem cells through altering the gene expression from a somatic cell. Studies on mice and rats found there to be advantages in cell differentiation and growth capacity with improvements in behavior and senses. Later studies found there to be multiple sources to subtract the stem cells for use. However, some studies failed to lead to improvements with disadvantages arising, including the formation of malignant tumours and immune rejection of stem cells. Therefore, more studies need to be conducted to understand tumour growth as well as the long-term effects. Neural stem cells are generally derived from fetal or adult brain tissue, however do provide a low amount of cells. There are two main types: endogenous cells which derive within the organism in the dentate gyrus of the hippocampus, the subventricular zone, and olfactory bulb. These cells function by producing neurotrophic factors to regulate the inflammatory environment and secrete factors which promote synaptic plasticity. Survival of the neurons are extremely low, therefore gene modification and direct injection of cytokines have been used to increase the survival rate. However, these have been concluded as insignificant, meaning more studies to ensure safety must be conducted. Exogenous cells are those derived outside an organism, such as from ESC, iPSC, bone marrow, and adipose-derived MSC. These cells can differentiate into neurons, astrocytes, and oligodendrocytes when induced by different factors. The advantages of these cells relate to the ability to target specific damaged areas of the brain to help in replacing damaged tissues, whilst encouraging the brain's own repair mechanisms. Disadvantages relate to immune rejection as well as ethical concerns surrounding embryo use and major operations on the adult brain which have high risk factors. Mesenchymal stem cells are multipotent cells which are easy to obtain and grow from multiple sources. These cells, when injected, locate the injured site, slow apoptosis processes, increase fibroblast growth, and stimulate endogenous cell growth. Studies on rats injected with these cells have shown there to be a decrease in infarct size. This is thought to be due to MSC producing and secreting substances to reduce inflammation and stimulate self-repair. The infusion of MSC intravenously into patients in clinical trials saw no adverse effects and improvements in modified Barthel index and increase in neuroplasticity. Further clinical studies have been undertaken on patients receiving MSC injections where treatment was deemed safe with no side effects but no clear evidence in improvements or benefits. Further research should be aimed towards finding the correct dosage amount, the best time for intervention, the most appropriate route of administration, gene modification to improve the survival rates of the cells, and finally cell homing, whereby the cell targets the exact specific target they need to be.